Welcome to my channel. Today we will discuss about disaccharides and polysaccharides and osazone formation. Disaccharide, it's nothing but that the combinations of two monosaccharides like glucose fructose, fructose galactose, fructose mannose. So two monosaccharides joined to call disaccharide. We will discuss about today sucrose. Sucrose is also one of the disaccharide sugar. Sucrose is a disaccharide and meaning it is made up of two monosaccharide units, glucose and fructose, linked by glycosidic bond, and also the this sucrose having the some chemical formula. It's a C12H22O11. So normally the sucrose it will be occurs in many plants, especially in sugar cane, sugar beets and fruits. The sweetened taste of sugar, sucrose, it's commonly used as table sugar due to its sweet taste and is widely used in cooking and food industries. The energy sources provides a quick source of energy as it's broken down into glucose and fructose during digestion. Non-reducing sugar, sucrose is a non-reducing sugar like other than sugars, it's called as a reducing sugar because of it does not react with the benedict solution. The key point, a carbohydrate identification test. Hydrolysis reaction, when hydrolyzed like a acid enzyme, they like a sucrase, always the ASE end word, it's called as a enzyme. It breaks into glucose and fructose. So, what is the role of sucrose? The sucrose, it's a main primary transport sugar in plants moving through the phloem from sources like a leaves to the roots. And commercial production, they are extracted and refined from sugar cane and sugar beet in large scale sugar industries. Dental and health effects, excess sucrose intake is linked to teeth decay, obesity, diabetics if consumed excessively. Example, if you are mix the sucrose solution with the Benedict reagents and eat it, there is no color, it will be changes because it's unlike glucose. This is because sucrose is a non-reducing sugar. Except the sucrose, if you are adding any type of the sugar solution in the Benedict test, we are getting there any colors, but this sucrose solution will not get any colors because of this is or the non-reducing sugar. And this structure show that the sucrose that is nothing but it's a combinations of glucose and fructose. Glucose is one of the monosaccharide, fructose is another monosaccharide. So both of them structures having the C6H12O6, but the C6H12O6 of the glucose having the first carbon atom above the aldehyde group, the fructose structure having the ketone group. And these both of the sugars they combine by the bonding glycosidic bonding. Yes. So next we will move on to the lactose. Lactose is also one of the disaccharide. This lactose, it's made up of two monosaccharides like a glucose and galactose. Just I remember, sucrose is a combinations of glucose and fructose. And chemical formula, it's the chemical formula of the lactose C12H22O11, same as sucrose, but the components linkages are different. Okay, so all the formula is C12H22O11, but the combination is differ. So lactose always they found in where means that is like a milk. It's a main sugar found in milk and dairy products. They like milk related products. So that's why the lactose is called as another name milk sugar. Yes, already you know that is a sucrose. It's need for the sucrase enzyme and also similar to the lactose. It need for the lactase enzyme. They are breakdown of the galactose. It will be productions of the glucose and galactose. All the things they are occurs in the small intestine. Okay. Yes. So, and come back to the reducing sugar. The lactose is a reducing sugar, meaning it gives a positive result in Benedict test. It's always the color changes from blue or bring red or sometimes they have developed to the orange color. So, based upon the color development, they are presence of reducing sugar or not. Okay, so the lactose it will getting the result. The color changes from blue to bring red color they called as a reducing sugar. And one more thing, the bonding is like a beta 1 comma 4 glycosidic bond. The glucose and galactose in lactose are joined by beta 1 comma 4 glycosidic linkages. Lactose intolerance, many people lack enough lactase enzyme, leading to lactose intolerance, causing bloating, gas, and diarrhea when consuming dairy products. So, most probably, they have the intake of milk contained product. They need for the some type of the enzymes, they like a lactase. Sometimes these lactase enzyme we will lacking means they are the lactose, it will be increased in our body. Okay, so they maybe leads to the diarrhea. So, what is the energy sources? Lactose provides energy after digestion and is especially important in infants. Used in pharmaceuticals, lactose is used as a carrier or filler in tablets and capsules due to its stability and solubility. Microbial fermentation, lactose is fermented by bacteria to produce the lactic acid, which is important in making yogurt and cheese. The example, if you add Benedict solutions to lactose solution and heat it, you will observe a color changes green to red, confirming that lactose it acts as a reducing sugar. Yes, this structure is showed that the lactose structure, lactose it's nothing but that is a disaccharide. It's a combination of galactose as well as a glucose. Both of them they having the same like a C6H12O6 the molecular formula, but the linkage is beta 1 comma 4 glycosidic linkages. Yes, so next we will move on to the osazone formation. Yes, already the osazone formation, the main purpose they are identifications of finally. So what type of the sugar will be present, they based upon the sugar will be confirmed with the help of microscope in the shape. Okay, under the microscope only they identified the what type of the sugar. Yes, so reaction with the phenyletrazine, once they are sugar will be there means they are reaction with the phenyletrazine. Okay, so the osazones are formed when reducing sugar reacts with the excess phenyletrazine under heat. So only the heat conditions they are mixed with the, they are combined with the sugar as well as the phenyletrazine reagent. This reaction specifically targets the carbonyl group. Why? Because means always the carbonyl group like any functional group will be present. Already I told that the glucose is having aldehyde group in the first carbon atom and also the ketone group it will be present in the second carbon atom. The identification of sugar, osazone. So, reaction with phenyletrazine. Osazone are formed when reducing sugar react with the excess phenyletrazine under heat. This reaction specifically targets the carbonyl group, aldehyde or ketone. Identification of sugars. Osazone crystals have distinct shape, needle shapes for glucose and fructose. Sometimes they are broomstick shape, which help in identifications and differentiation of sugars under a microscope. Always the phenyletrazine reacts with the sugars. First initially they are disturbed with the carbon group like a C1 as well as a C2 group. Always the functional group will be present in this carbon atoms. Like sugars like glucose, the first carbon atom having the aldehyde group, second carbon atom having the second group carbon atom like a fructose having the ketone group. Examples of osazone crystals: glucose, fructose, mannose, needle-shaped osazones, lactose, powder puff and hedgehog shape, maltose, sunflower shape. Diagnostic tool in the past. Before modern techniques, osazone formation was a classical method used to identify sugar in clinical and research lab. Yes, so this reaction it's show that the how to they are react with the phenyletrazine from the sugar. So like first stage we will take the aldose, one of the sugar, they are react with the phenyletrazine. Initially they are liberated to water molecule. So that is the first step, we are getting the result phenyletrazone. So the first aldose of oxygen they are reacts with the phenyletrazine of H2, they are liberated to water molecule, they finally productions of phenyletrazone. So the second stage, again the phenyletrazone reacts with one more phenyletrazine Again, they are liberated ammonia and also the C6H5NH2. So intermediate step will be formed. Again, they are because of phenylhydrazine reacts with the equal amount of the sugar molecule. They are equal amount of the sugar phenylhydrazine. So first initially the phenylhydrazone will be formed. Finally we will get the precipitation. So that's why they needed for one more time of the phenylhydrazine. So finally after the intermediate steps of the phenylhydrazone, they are again reacts with the phenylhydrazine to liberate the water molecule similar to the first step. Finally they are productions of osazone. Okay, these are the osazone reaction. So first stage, it's nothing but it's a reacts with the what is that? Aldose reacts with the phenylhydrazine to will be developed into phenylhydrazone. Again that phenylhydrazone, the second stage reacts with the phenylhydrazine, ammonia as well as the C6H5NH2 will be liberated and the intermediate steps of the phenylhydrazone will be developed. After the phenylhydrazone reacts with the phenylhydrazine, liberate the water molecule, they are finally developed to the osazone. Yes, so next we will move on to the starch. Starch is nothing but what? That is, it's nothing but that is a polysaccharide. Starch is most widely distributed in vegetable kingdom. In nature, it is transformed into complex polysaccharide like gum and cellulose. Always you know that starch will be present in the where? It's a plants. Okay, and into a simpler mono and disaccharide by enzymes working in vegetable kingdom. So always there sources of starch: potatoes, wheat, maize, as well as the rice, barley, and root. It is interesting to note that no two sugars to give a identical starch because of they have its combinations of more than 10 monosaccharides. It's called as a what is that? Polysaccharide. Yes, what is the structure? Starch is a polysaccharide and made up of glucose unit C6H10O5 overall N. It is a primary storage carbohydrate in plant stored in seeds, tubers, roots. It's composed of two components, amylose. Like always, the amylose 20 to 30% linear and branched polymer of alpha D glucose. They are linked by alpha 1,4 glycosidic bond. Always they forms a helical structure insoluble in cold water. And the next one, amylopectin, always the amylopectin 70 to 80%. It's a highly branched polymer. When compared to amylose, it's a unbranched structure, but amylopectin, it's a highly branched polymer. As a alpha 1,4 bonds in chains and